Good morning, everybody. Welcome to our lesson on viruses. I so wish I could be teaching this in front of you right now. Timely topic. 66% of you voted to have the viruses lesson come first, so that's what we're doing. This is really just a single day lesson, so we're going to pop through some different things, how viruses affect prokaryotes versus eukaryotes, and we will be looking at what a virus is and how it functions. Okay, so first up, what is a virus? It's an intracellular parasite, and it has two parts to it: genetic information and a protein coat. So that protein coat is called a capsid. It surrounds the genetic information inside the virus. That's it. A piece of bad news wrapped in a protein. So let's take a look at some common ones. Here's the tobacco mosaic virus. This would affect plants. Tobacco is a plant. It's an RNA virus. And here's its protein coat, its capsid. Adenoviruses, common cold type of viruses. These are your rhinoviruses that affects anything that's upper respiratory system. Has glycoproteins on the surface. Its capsid is out here in purple, and you can see that across the way. So everywhere you see the purple, that's the capsid. And adenoviruses are DNA viruses. If we look at influenza viruses right here, they're a little bit different. So they also have glycoproteins, lots more of them on the surface. They are RNA viruses, and they may also have a lipid coat on the outside. So that membranous envelope here is a lipid coat, and you can see it's capsid on the inside of the virus. And then finally we have our bacteriophages, which we've learned about before, and they only infect bacteria, prokaryotes. So they are DNA on the inside of its head. Its capsid is here, and they have a few other parts to it too, so we'll be going through those as well. All right, since they can't carry out their life cycle independent of a host cell because they're not made of cells, viruses are not traditionally considered to be alive, as if that matters, right? So in order to be considered a living thing, you have to do all of the life processes. However, viruses only do one. They reproduce, that's it, and they need a host to do so. So they were first discovered by Dmitry Ivanovsky, who was studying the transmission of the tobacco mosaic disease. They knew something smaller than cells was actually causing that disease. So let's take a look at what they did. So they extracted some sap from tobacco with the disease, and they passed it through a filter, which is known to trap the bacteria. And then they took that and they rubbed it on healthy tobacco plants, and sure enough, the healthy plants became infected. So relatively simple experiment. Here's what the tobacco mosaic virus looks like. It's got its protein coat on the outside. The red represents the RNA on the inside. So there are some others that you may be familiar with. This is more of a eukaryotic virus. It's the herpes simplex virus. That's cold sores. Measles are a virus. Polio and smallpox. The difference for these is they all have a vaccine towards them. So as long as you're vaccinated, you're protected against those viruses. A few others. So 2009, there was a pandemic for H1N1 influenza A. That was the swine flu. There was a screening for this, and this should look familiar because we're seeing a lot of this today. And then 1918 flu pandemic, this was called the Spanish flu at the time, killed millions of people. All right, so what we're going to do is look at the differences between prokaryotic viruses and eukaryotic. We'll be doing prokaryotic viruses first, and please, please, please keep in mind that these viruses only infect bacteria. That is it. You are not a bacteria, therefore you cannot be infected by a bacteriophage. All right, so Prokaryotic viruses are also known as phages, which come from their habit of eating lawns of bacteria on petri dishes. And there are 10 to the 31st phages on the planet. That is pretty incredible. Okay, so let's take a look at what a lawn of bacteria looks like. First up, this is when we look at the phages themselves, it's kind of a cool fun fact. There's a larger number of bacteriophages than there are all of the stars in the universe. So it's pretty neat. Here's a T4 phages infecting E. coli is the bacteria, and you can see all of the viral bacterial phages infecting it. And this is a petri dish. So when we have lawn of bacteria, it means that there's so much bacteria, it shows up like a grass lawn on top of the nutrient agar. However, when we look at this, we can only see the individual colonies that are left over. So any other space in between here means that the phage infection killed those bacteria, and so they're dead, they don't appear anymore. All right, there are two kinds of bacteriophages, the virulent phages and the temperate. And it's all about the timing of the viral life cycle. So we're going to look at the lytic cycle first. This is the classic viral life cycle. The stages are infection, synthesis, assembly, and release. The assembly is autocatalytic. I'll explain what that means in a minute. An example of this is the T4 phage or the lunar lander. So let's pull this up. We've got our attachment here, right? You see the bacteriophage landing on the bacterial cell. Then you have, here is the injection of the viral DNA, right? This is the infection portion. So it's injecting its DNA inside of the bacterial cell. And then you've got synthesis of the viral genomes and the proteins on the inside of the cell. You have autocatalytic assembly. So that means it just automatically assembles into new bacteriophages, and when they are fully assembled, they will burst out of the bacterial cell, killing the cell, and they will release into the environment over to infect other bacterial cells. Okay, so that's called the lytic portion. If we look at the assembly portion here, you can see how these are built. So you've got your head here, DNA is on the inside, and you can see how the whole thing is put together into this little lunar lander. Okay, so infection, synthesis, assembly, release. It's fairly rapid. Okay, so this is a quick, in terms of timeline, the lytic phase is very quick. If we compare that to the temperate phages, this is the lysogenic cycle. So here, it just takes a longer period of time, and it's usually due to environmental considerations, such as the number of available bacteria to actually infect in the environment. So if you recall, virus needs a host. If there's not enough bacteria, the population density of bacteria is very low, then you will see more of a lysogenic cycle before it goes lytic. Okay, so phage DNA splices into the bacterial chromosome. When the cell replicates, the phage DNA is replicated as well, and it can go lytic as conditions dictate. Okay, so let's go through some of that. An example of this would be the lambda phage. So I want to pull up the picture large. Okay, so this is what we looked at before. Here's your classic lytic cycle that we just went over. Phage injects its DNA. If the lytic cycle is induced, you'll have synthesis, assembly, and then you have release. Okay. However, when we look at the certain factors that determine whether it's lytic or lysogenic, it usually has to do with the population density of the bacteria themselves. That meaning, if the population density of bacteria is very low, then it will be a lysogenic cycle instead. Okay, so we can take a look at how that works. Here's the phage DNA. So, the bacteriophage still has to land, just like this. And when it injects its DNA, it's incorporated into the bacterial chromosome. So as the bacterium reproduces, it will also copy the viral DNA as well. transmitting it to all daughter cells, right? This will go through mitosis, and then the cell divisions produce a population of bacteria infected with the prophage, which we can see. So this is showing the daughter cell with the prophage. And then occasionally the prophage exits a bacterial chromosome, and it will initiate a lytic cycle. Okay, so once that population density of bacteria is very high, then it will induce the lytic cycle, and boom, there you go, you have infection of the bacteriophages, which will be released to infect other bacteria in the population. Okay, so that's lytic versus lysogenic. Phage therapy has been around for a really long period of time, however, not really in the US, mostly because we have very strict laws from the FDA. So it's still considered a new avenue of research because we are running out of antibiotics that kill bacteria, and so this may be an interesting way of potentially creating some methods of killing bacteria without the use of antibiotics in the future. Right, so here's a phage plaque on a colony of Bacillus anthracis, right? And you can see up close, they would be attacking the bacteria. Okay, so now we're going to get into the eukaryotic viruses. Eukaryotic viruses are much more diverse than the phages. They can have DNA or RNA genomes. They're single-stranded or double-stranded, and many have a lipid envelope, that membranous envelope that surrounds the protein coat, and that makes them much more different and much more complex. Okay, so we're going to take a look at the differences in how these DNA or RNA genomes work. So we'll look at the DNA virus first. Here it is. You can see the DNA inside the capsid, right? So there's entry into the host cell, and you can immediately see that the capsid disperses. Here's the viral DNA. There will be replication of the viral DNA. The mRNA from this DNA, from the viral DNA here, will be used to create more capsid proteins, right? Protein synthesis and the process of transcription and translation. And then there'll be self-assembly of the viral particles with the DNA on the inside and the capsids, and then they will burst from the cell. If we look at an RNA virus over here, it's different. Okay, so you can see the envelope. Here's the lipid envelope. Right, here's its capsid on the inside, and there's the RNA, the genetic material. So you have all these glycoproteins on the surface, and in order for an RNA virus to get inside the cell, these glycoproteins have to match up with a receptor on the surface of the host cell in order to gain entry. If it does, it will enter, and now you can see the capsid with the viral genome. It is an RNA virus, which will act as a template to produce the glycoproteins, which we can see here. Here's your ER. Here are the proteins that are produced, being carried in a vesicle to the surface of the cell membrane. Transcription of this RNA will take place. There'll be a copy of the genome. We have assembly, and then the virus will leave through the cell membrane. It will burst forth. Usually millions of viruses will do that. To go on to infect other cells. Okay, so that's the difference between DNA and RNA viruses. Right, so if we kind of pop through these quickly, there are lots of different classes of animal viruses. Right, these are all eukaryotic. You can have double-stranded DNA viruses. The adenovirus, respiratory, can be tumor-causing, does not have a lipid envelope. Papavirus, that's a virus that causes warts and some cervical cancer. Herpes virus does have a lipid coat. That's herpes simplex 1 and 2, the cold sores versus the genital sore version. Shingles or chickenpox, and then the Epstein-Barr virus. Pox viruses also have a lipid envelope. That's smallpox and cowpox. Single-stranded DNA viruses, this is parvovirus, does not have a lipid envelope. Parvovirus actually can infect dogs. And then you have double-stranded RNA viruses, the reovirus, that causes some diarrhea and Colorado tick fever. Sorry. Okay, So there are some single-stranded RNA viruses that serve as mRNA. So an example of this is the rhinovirus, a common cold, Hep A, and other intestinal viruses. Here's the coronavirus. Coronaviruses have been around for a while now. They do have a lipid envelope, and one of the first ones caused SARS, or severe acute respiratory syndrome, that is different from what you are seeing today. Flavivirus, yellow fever, Hep C, togavirus, this is rubella, and you should be vaccinated against that, okay? There are some single-stranded RNA viruses that act as a template for mRNA synthesis. This is going to be the Ebola virus, some influenza viruses, measles, mumps, and rabies. And then finally, we have retroviruses, which we'll take a close look at in a minute, that cause HIV/AIDS. All right, so let's go into some examples of animal viruses. We'll look at influenza first, followed by HIV and how a retrovirus works. It has a special enzyme to it. I've added in COVID-19, so we'll talk a little bit about that, and then some plant viruses. Okay, so first up is influenza. It is an RNA virus, easily transmitted in aerosol form, right? So that means by coughing or sneezing, it's in an aerosol form. If you're close by, you can easily inhale this RNA virus. So I think most of us are pretty familiar with the symptoms of influenza. Here they are: some headache, usually high fever, you can have muscle joint pain, respiratory issues with coughing, can be gastric. Sometimes people vomit with the flu, runny nose, or could be total congestion, sore throat, and then aches. So it's responsible for more deaths in human history than any other known virus. This picture was from the 1918 influenza pandemic. It mutates rapidly. So we call this antigenic shift, meaning the surface antigens, aka the cell receptors on the surface, can mutate rapidly. So permanent immunity to influenza is not possible. This is why each year they use modeling to predict, to create the flu vaccine for that year, for whichever strain they think is going to show up. All right, so when we look at antigenic shift, you can have a highly pathogenic avian strain, a bird flu. Okay, we can see its receptors on the surface. This pink one is similar to the human strain over here. However, the human strain is green, so we can see the different colors showing the different glycoproteins on the surface. In a new highly pathogenic strain here, you can see the mix between the two. And so when we say there's crossover from one species to another, this is how it occurs. And there can be co-infection by two flu strains that trigger a large-scale viral recombination. And so that recombination is due to the glycoproteins on the surface of the virus. Okay, so there are influenza A subtypes, and we can track this through time. So this shows us a timeline. So the current thinking is that the H1, and I'll explain what the H's and the N's mean in a minute, showed up late 1800s, possibly an H3 strain here. But here's our first recorded 1918 strain as H1N1. Here's a mutagenic shift to H2N2. We can also see that shift here. Here's H1N1 again. However, this H1N1 crossed over from pigs. That was the swine flu. And then we have H3N2. So what comes forward? Not really sure. It could be an H3N1. Might be the next shift, and they'll use modeling to predict that. Okay, so here's how viruses are named, right? We look at the virus type. So this would be an influenza A, where it came from, Fuji, the strain number, the year it was actually isolated, and then the virus subtypes. So these names, H1N1, H3N2, come from the type of glycoprotein on the surface. So you can see this one here is neuraminidase 2. Here's the 3, this little rectangular one right here. Okay, so the H stands for hemagglutinin and the N for neuraminidase. All right, I can't show you that. I will put the link up. There's a copyright infringement for NPR. I can't show that video to you, but it's actually pretty cool for you to see. So you can get a picture of how these viruses actually infect humans on a respiratory level. All right, so we're going to look at HIV next. This is human immunodeficiency virus. It is a retrovirus. The genome is RNA. The virus has a code for reverse transcriptase. Okay, so that is an enzyme that allows the RNA virus to make DNA from the RNA. Okay, so that's a central dogma violation. The central dogma states DNA to RNA to protein. However, retroviruses like HIV have a special enzyme that allows them to make a DNA copy of the RNA genome, and then that can be spliced into the host cell chromosome. Okay, so just if you recall how this works, here's the glycoproteins on the surface. This is an RNA virus, gains entry into the cell. Here's the host cell, here's the viral RNA, and it's got that special enzyme. So there'll be an RNA-DNA hybrid at first. Here's the DNA inserted into the host cell genome. So this occurs with HIV-positive individuals if the virus gains entry into their T cells, their helper T cells, which actually act as the alert system for the rest of your immune system to create antibodies. Okay, so we can take a look at actual footage of, here's the membrane of the white blood cell here, right? This is of the T cell, the helper T. Here's HIV entering the cell, and then you can see it exiting over here, right? And it will go on to infect other helper T cells in the area. So the acute infection when people are first infected with HIV is actually very similar to the flu, so many people think that they have the flu at the time. As the virus destroys immune system cells over the time, then the body can become prone to opportunistic infections. So that's all based on the T cell count. So if that helper T cell count drops below 200, then the person is considered to have acquired immune deficiency syndrome, which makes them susceptible to other types of infections that you would not normally get with a functioning immune system. So the virus can be spread through bodily fluid exchange, and you can see the prevalence of it in the world, with the southern African countries having the highest prevalence. Okay. Oh, sorry. I feel like we're in class with me clicking different things. Okay. There are various treatments primarily available in developed countries, and mostly because we have access to good health care and health insurance. You can see how expensive it is. Just one of the drugs to help with treatment is $1,200 a month, and it's probably even more now. All right, so here's coronavirus disease 2019. As we sit home doing remote learning, COVID-19, you can see where the name comes from. The CO, the VI, here's the disease, and it was discovered in 2019. So currently in the United States, this is all from the Centers of Disease Control as of 3/26. The United States is in an acceleration phase of COVID-19. It's a beta coronavirus, which does have its origins in bats. The complete picture of COVID-19 is not known yet, and so that affects how we approach it and what treatments are available. Reported illnesses can range from mild to severe. So at this time, mostly older adults, ages 65 plus, and individuals at higher risk. So higher risk can mean people with diabetes or heart disease or other lung issues that make them at increased risk, pregnant women as well. However, with social distancing, as we're all practicing right now, the immediate risk is low. However, people in places with ongoing community spread can have increased risk of exposure. Okay, so how COVID-19 spreads. Coronaviruses are a large family of viruses. Some cause illness in people, or in canines, or in felines, or as the news reported yesterday, there is a tiger that's been infected in the Bronx Zoo. So we can see a transmission from humans to animals as well. Rarely, there are animal coronaviruses that have emerged to infect people. So MERS, Middle East Respiratory Syndrome, was another example of a coronavirus, as well as SARS, Severe Acute Respiratory Syndrome, that occurred in Asian countries. That was back in 2009, I believe. So currently, we're in this acceleration phase, and the duration and severity of a pandemic like this can vary depending on the virus characteristics and the public health response to it. So when we say community virus, that means that people have been infected and it's not known how or exactly where they became exposed to the virus. So more than half of the US is seeing some level of community spread. All 50 states have reported positive cases though. All right, so what we know, many of the patients at the epicenter of the outbreak in Wuhan, China had some link to large seafood and live animal markets, suggesting an animal-to-person spread at first, but then later on, a growing number of the patients reportedly did not have exposure to the animal markets, and that indicates a person-to-person spread or a community spread. Okay, so the complete clinical picture of COVID-19 is not fully known at this time. Reported illnesses can be very mild, that's some with no symptoms at all, to severe, which is mostly in older individuals at this point, resulting in death. Information so far, right? We have to emphasize that so far suggests that most COVID-19 is mild, but a report out of China suggests serious illness occurs in 16% of cases, and we're starting to see some other comorbidities such as obesity with other issues such as diabetes or heart disease puts people at greater risk for severe symptoms and severe illness. Okay, so there's some great information on the CDC website as far as frequently asked questions, and if you have any questions for me, I'll be happy to answer those as best as I can. And we can take a look at the virus, right? Typical eukaryotic virus here. These are the glycoproteins on the surface. And it is an RNA virus. It does have a lipid envelope as well, so its capsid will be on the inside here. Okay, so we can look at its a few things here. Here's the membrane protein, the M protein there. That's the central organizer. It determines the shape of the viral envelope. Here's its RNA in the center. We've got its nucleocapsid protein, the N protein on the inside, right, bound to the RNA genome, right? So that's going to make up its capsid protein coat. And then you've got the spike protein on the outside, critical for binding to host cell receptors, so that's how it gains entry into cells. And then the E, the envelope protein, interacts with the N to form the viral envelope or the lipid envelope on the outside. All right, then there are plant viruses. Plants can get viruses too. They typically manifest as blotchy pigment patterns. You can see that in flowers and in some fruits and vegetables, and that's all I really have to say about that, but plants can get viruses as well. All right, so that's basically the difference between our prokaryotic and eukaryotic viruses. We are going to take a quick look at some things that are not viruses, but are infectious particles, okay? So what a virus isn't, and these are some of the simplest infectious particles known. So you can have a viroid. It's a disease-causing RNA molecule. This is only in plants. So the RNA sequence would be in the potato, inserted into its genome, and it does not code for any protein at all. So that's called a viroid. You can have prions. Now, these are disease-causing protein molecules. They're basically misfolded proteins. So prions are kind of scary. And they have no genetic material, so it's just a protein. But as a misfolded protein, what happens to them is that they actually aggregate, or they stack up on top of each other like this. And in cases of BSE, this is the mad cow disease. That's called Creutzfeldt-Jakob disease in humans, but it can also affect sheep and deer as well. These aggregates of prions form in the brain, and then it destroys brain tissue, hence mad cow disease. Okay, so you get this characteristic spongy form plaque in brain tissue of a person with mad cow disease, and it's not something you want. It's putting holes in your brain. Okay, so originally that mad cow disease showed up in the UK in, I want to say it was the late 90s, early 2000s, not exactly sure of the date, but they had some cows that were infected with it. Prions are not something that you can destroy through heating or cooking, so people had eaten the tainted meat from those cows and were infected with the prions and then died from that disease. So it caused a massive recall of all the meat at that time. All right, so that's what a virus isn't. Okay, things that you're responsible for. You should be able to explain why viruses should be considered living or non-living. Explain how different types of viruses utilize the information systems of cells to complete their life cycles, right? So in prokaryotes, that could be the prophages in the lysogenic cycle, also the differences between RNA and DNA viruses. You should be able to compare the bacteriophages with the eukaryotic viruses, right? Eukaryotic viruses, much more complex, and certainly know the difference between the lytic and lysogenic cycles of the bacteriophages that infect prokaryotes. Explain why some eukaryotic viruses have RNA genomes. Explain the features of the viral life cycle of a typical DNA, RNA, and retroviral eukaryotic viruses. Consequences of viral life cycles on human health, right? And so those are some of the diseases I highlighted, like influenza, HIV, and COVID-19, and then compare viruses to other infectious particles that have been described by science. That's the viroids and the prions. So I hope that was helpful. Let me know if you have any questions. You can email me or hit me up through the Remind. Talk to you soon.